So this is a very interesting work. We know venetoclax is an important drug in T11-14 myeloma, and in combination with Velcade, it works beautifully well, even in non-11-14 patients who are overexpressor of BCL2. However, there has been data that has not been as clear in regards to overall survival in the Bellini study. So we have investigated in this particular research work what the resistance to venetoclax means, why do patients not do so well afterwards possibly, and what can we do to improve the outcome. And if I give a very brief rundown on what we did was that we developed a venetoclax resistant cell line, and we developed multiple clones. It was single clone. And what we found was that when cells develop venetoclax resistance, they are also resistant to a number of traditional myeloma drugs. That includes proteasome inhibitor, Velcade and carfilzomib. It includes immunomodulators, pomalidomide, lenalidomide. It includes all the cytotoxic drug, melphalan, bendamustine, cytoxan. So it's a possibility that when you develop venetoclax resistance, you may not respond much to the subsequent treatment. So that's one part of the story. The second part and very exciting part is that even though the response to this agent is not good, when we use immune targeting treatment, and why did we look at those is because immune targeting treatment goes through different pathway in killing the cells, not through the BCL2, the typical apoptosis pathway. So we reasoned that if you use a different pathway, maybe we can kill. And that's exactly what we saw. That if you look at the ADCC daratumumab mediated killing, that's very good. If you look at a CAR T cell mediated killing, it's excellent. And we even had a patient who was venetoclax resistant. We took his bone marrow. It was sensitive to CAR T cell. Patient actually got CAR T cell and had a beautiful response. So the bottom line is that if you are resistant to venetoclax, there is a chance that you will be resistant to other traditional myeloma drug, but your immune treatments, including daratumumab and other antibodies, may still be effective, and that's how we may need to sequence them to get the optimum response.